There are three major sections of the ear. First is the outer ear. This is the visible portion of the ear that collects and directs sound waves toward the tympanic membrane. Second is the middle ear. The middle ear is the space between the eardrum and the inner ear that contains the three auditory ossicles, which convey vibrations through the oval window to the cochlea. Finally, we have the inner ear. The inner ear is a liquid-filled membranous portion of the ear involved in hearing and balance. At the start of this process, sound waves enter into the ear through the external acoustic meatus and travel down the auditory canal, where they come in contact with the tympanic membrane, also known as the eardrum. The tympanic membrane vibrates, and vibration is transferred to the bony ossicles. The bony ossicles are the malleus, the incus, and the stapes. The bony ossicles and the tympanic membrane work together to amplify sound. The stapes works by using piston-like movements which sends vibrations into the bony labyrinth. Due to the movement of the round window membrane, the stapes movement can displace the perilymph, which allow vibrations to enter the labyrinth. Vibrations that are produced by the stapes are then sent through the spiral structure or the cochlea, allowing for the sound waves to meet the round window. The cochlear duct is positioned in between the ascending scala vestibule and descending scala tympani. The cochlear duct is filled with endolymph fluid. The actual nerve impulses that are sent to the brain via the cochlear nerve are generated by special cells called hair cells, or stereocilia, that are covered by tectorial membrane. Both endolymph and perilymph are found surrounding the hair cells of the inner ear. Endolymph, high in potassium, is found around the apical surfaces of hair cells, while perilymph, high in sodium, is found in the sides and bases of the hair cells. Here is an illustration of the depolarization of the stereocilia cell. Vibrations of the endolymph causes vibrations in the basilar membrane. This moves stereocilia to the tips of the hair cells against the tectorial membrane and open potassium channels in them. The influx of potassium from the endolymph depolarizes the cell. Depolarization of the hair cell causes the release of a neurotransmitter at its basal surface of the initiation of nerve impulses in a sensory neuron that synapses with it. These impulses travel back along the auditory nerve and up to the brain.